Good morning, students. Today we are talking about structures and functions of reproductive organs in mammals. Our focus is on mammals, but as we go on talking about reproduction generally, we are going to be mentioning one or two animals that are not mammals in the process. What is reproduction? Let's start with that. Reproduction can be defined as a process whereby living organisms give rise to young ones of their kind, or whereby living organisms give rise to offsprings of their kind. When we say of their kind, a dog will not go and give birth to a fish, and a goat is not going to give birth to a human being, to a man. So it has to be of their kind. Also, a human being, a man or woman, is not going to give birth to a frog. That is why we said reproduction can be defined as a process whereby living organisms they give rise or give birth to offsprings or to young ones. Not just young ones, but the young ones must be of their kind. The reproductive structures of many animals are very similar, even across different lineages, in a process that begins with two gametes. So two gametes are involved. The two gametes are the egg and the sperm. And it begins with two gametes, egg and sperm, and ends with a zygote. A zygote is a fertilized egg. In animals ranging from insects to humans, male produce sperm in testes, and sperms are stored in the epididymis until ejaculation. Sperm are small, mobile, low-cost cells that occur in high numbers. The sperm are very numerous when they are produced. The reason is because the sperm cannot live for long. And another reason is because the sperm have to swim up into the vagina and to the womb to go and meet with the egg. So the sperm are very many. In fact, there are millions of sperm at a single ejaculation, such that only one of it will fertilize the egg. And that is why it is said that every human being is a winner, even from birth, because millions of sperm was released by the male or father, and only one of it was able to meet with the egg or with the ovum and to fertilize it. The remaining died. So that's why sometimes it's said that everyone is a winner, even from conception. So the sperms are stored in the epididymis until ejaculation. Sperm are small, mobile, low-cost cells that occur in high numbers. Females produce an ovum or egg, and the ovum or egg matures inside of the ovary. Eggs are very large cells that require a substantial investment of time and energy to form. Eggs are non-mobile and are rare relative to sperm. The eggs are not many. One egg is produced per month. So one egg in January, one egg in February, one egg in March, one egg in April. So they are produced about 28 days or thereabout. When the eggs are released from the ovary, they travel to the uterine tubes for fertilization. In animals that produce through internal fertilization, fertilization takes place inside of the uterus. But there are some animals which are not mammals that practice external fertilization. So the eggs are released into the aqueous environment, and the animals they go there and pour the sperm on it and fertilize it. All sexual reproducers have females with ovaries that produce large eggs, which subsequently travel down a uterine tube, and males with testes that produce small, plenteous sperm stored in an epididymis. Of course, beyond this general anatomy, there are some interesting differences in different types of animals. In some invertebrate species, including many insects and some mollusks and worms, the female has a spermatica, a specialized sac, which stores sperm for later use, sometimes up to a year. Fertilization can be timed with environmental or food conditions that are optimal for offspring survival. Non-mammalian vertebrates, such as most birds and reptiles, have a cloaca, a single body opening which functions in the digestive, excretory, and reproductive systems. Mating between birds usually involves positioning the cloaca opening opposite each other for transfer of sperm from male to female. Dogs are a rare exception, where the males have a penis. Mammals have separate openings for the systems in the female, and placental mammals have a uterus for support of developing offspring. The uterus has two chambers in species that produce large numbers of offspring at a time, while species that produce one offspring, such as primates, have a single chamber. Now we'll move on to the structure or the anatomy of the mammalian reproductive organs. We'll look at gametogenesis, and also we'll look at hormonal regulation. Gametogenesis is the production of sperm and eggs. The sperm and the eggs are the gametes. So gametogenesis is the beginning of the gametes, and the beginning of the gametes is the production of the gametes. So gametogenesis is the production of sperm and eggs, and takes place through the process of meiosis, M-E-I-O-S-I-S. It takes place through the process of meiosis, and meiosis produces haploid cells with half the number of chromosomes normally found in diploid cells. Hormones are dynamic in these, so this process can be trickier to understand than basic anatomy of gametogenesis. Hormonal changes are the center of fascinating biology of reproduction. The human female and male reproductive cycles are both controlled by the interaction of hormones from the hypothalamus and interior pituitary with hormones from reproductive tissues and organs. The hypothalamus sends a gonadotropin releasing hormone, GnRH, to the anterior pituitary, the follicle stimulating hormone, FSH, and the luteinizing hormone. Follicle is FOLLICLE, while luteinizing is LUTEINIZING. So, follicle stimulating hormone and luteinizing hormone are released from the anterior pituitary into the blood as a result. Although follicle stimulating hormone and luteinizing hormone are named after their functions in female reproduction, they are produced and play important roles in controlling reproduction in both sexes. Now we look at how the female reproductive organ looks like, the anatomy of female reproduction. A number of reproductive structures are exterior to the female's body. These include the breasts and the vulva. Internal female reproductive structures include ovaries, oviducts, the uterus, and the vagina. And these are shown in this illustration, which is this diagram. So let's take note of the diagram and learn how to draw the diagram. This diagram is a diagram of a major female reproductive organs. Image is modified. The next is that human females become capable of reproduction at sexual maturity, which follows puberty. During puberty, the hypothalamus in the brain signals the pituitary gland to produce two hormones, follicle-stimulating hormone and luteinizing hormone. In females, follicle-stimulating hormone and luteinizing hormone stimulate the ovaries to produce the female sex hormones, estrogen and progesterone. This results in the development of secondary sexual characteristics, such as breasts, and causes the ovaries to begin producing mature eggs. This table below briefly summarizes the major organs and their locations and functions of mammalian female reproductive anatomy. The first organ is the ovaries, and their function is that they produce and develop eggs. The second organ is the fallopian tube, also called oviduct. It transports eggs to uterus, acts as site of fertilization. The uterus itself supports a developing embryo, and the uterus is sometimes called the womb. The cervix allows passage between the uterus and vagina. The vagina receives penis during intercourse, acts as birth canal, passes menstrual flow. The breast produces and delivers milk. The breast produces milk and it delivers milk to the embryo or the young one that is produced. Ovaries are the site of egg development. Egg development occurs in structures called follicles, which are lined with specialized cells called follicular cells that surround the egg and promote egg development. During the menstrual cycle, which is called the menstrual cycle, a batch of follicular cells develops and prepares the eggs for release. At ovulation, one follicle ruptures and one egg is released, as illustrated below. The ruptured follicle, which remains in the ovary, is then called the corpus luteum, corpus luteum. This corpus luteum secretes hormones that prevent menstruation until the egg has had time to be fertilized. If fertilization and implantation in the uterine wall occurs, then the corpus luteum continues to prevent menstruation. If fertilization does not occur, then the corpus luteum degenerates and menstruation occurs. The oviducts of fallopian tubes extend from the uterus to the ovaries, but they are not in direct physical contact with the ovaries. The ends of the oviducts flare out into a trumpet-like structure and have a fringe of finger-like projections called fimbriae. When an egg is released at ovulation, the fimbriae help the egg enter into the tube and passage to the uterus. Fertilization, which is the union of sperm and egg, usually takes place within the oviducts, and the developing embryo is moved toward the uterus for development. It usually takes the egg or embryo a week to travel through the oviduct. Now, I want to talk to you about oogenesis, also called female gametogenesis. What is oogenesis? Oogenesis is double O, then genesis. It is the process of producing an egg cell, and it occurs in the ovaries. Egg stem cells, called oogonia, divide by mitosis to produce up to 2 million oocytes. Oocytes are precursors to the egg. The process of oogenesis begins while the female is still an embryo undergoing development. The oocyte starts the process of meiosis and then pause during meiotic prophase 1. Because this process occurs during embryonic development, this means that a female mammal is born with every single egg she will be able to produce during her lifetime already present in an immature form, however, in her ovaries. This situation is very different from males whose spermatogonia, the sperm equivalent to oogonia, do not begin producing spermatocytes, that means the sperm equivalent to oocytes, until puberty. The oocytes remain in meiotic prophase 1 until the onset of puberty, when a series of events can lead to egg maturation. The anterior pituitary hormones, FSH and LH, cause some of the follicles to begin developing and oocyte inside the follicle to finish the first meiotic division. After completing meiosis 1, the oocyte pauses again, this time during metaphase 2. The released oocyte will begin traveling through the oviduct, still arrested in meiosis 2. If the oocyte is fertilized by a sperm, it will finish meiosis 2 and undergo unequal cytokinesis, that's cell division, to produce a fertilized egg, that is an embryo, and another polar body. If it is not fertilized, the oocyte degrades without completing meiosis 2. One final point before we move on to hormonal control of oogenesis. When an oocyte undergoes meiosis, it produces only a single egg. Again, this is different from spermatogenesis, which produces 4 sperm from each spermatocyte. The oocyte divides unequally so that almost all of the cytoplasm goes into only one daughter cell, rather than evenly distributed into both. The smaller cell is called a polar body, and it normally dies. How does hormones control oogenesis? Hormonal control of oogenesis. Oogenesis is controlled by FSH, LH, estrogen, and progesterone. FSH stimulates development of egg cells that develop in structures called follicles, which are located within the ovaries. LH also promotes development and maturation of eggs and induction of ovulation. Estrogen is the reproductive hormone in females that assists in ovulation and regrowing the lining of the uterus. It is also responsible for the secondary sexual characteristics of females, such as breast development. Progesterone assists in endometrial regrowth and inhibition of FSH and LH release. These hormones together regulate the ovarian and menstrual cycles. That's these 4 hormones: FSH, LH, estrogen, and progesterone. All of them together regulate the ovarian and menstrual cycles. The ovarian cycle governs the preparation of endocrine tissues and release of eggs, while the menstrual cycle governs the preparation and maintenance of the uterine lining. These cycles occur concurrently and are coordinated over a 22 to 32 day cycle, with an average length of 28 days. The first half of the ovarian cycle is the follicular phase. Slowly rising levels of FSH and LH cause the growth of follicles on the surface of the ovary. This process prepares the egg for ovulation. As the follicles grow, they begin releasing estrogens. Estrogen levels increase over the course of the follicular phase as the follicles continue to develop. In the menstrual cycle, menstruation flow occurs at the beginning of the follicular phase when estrogen levels are low. When the follicles are only just beginning to develop, rising levels of estrogen then cause the endometrium to proliferate, that is to grow, replacing the blood vessels and glands that deteriorated on the end of the last cycle. Ovulation. Ovulation occurs just before to the middle of the cycle, approximately day 14, when the high level of estrogen produced by the developing follicles causes FSH and especially LH to rise rapidly, then fall. The spike in LH causes ovulation. The follicle which is most mature ruptures and releases its egg. The follicles that did not rupture degenerate and their eggs are lost. The level of estrogen decreases when the extra follicles degenerates. Following ovulation, the ovarian cycle enters its luteal phase, and the menstrual cycle enters its secretory phase, both of which run from about day 15 to 28. The cells in the follicle undergo physical changes, and produce a structure called a corpus luteum. corpus luteum, corpus luteum. This corpus luteum produces estrogen and progesterone. The progesterone facilitates the growth of the uterine lining and inhibits the release of further FSH and LH. The uterus becomes prepared to accept a fertilized egg should fertilization occur. The inhibition of FSH and LH by progesterone prevents any further eggs and follicles from developing. The level of estrogen produced by the corpus luteum increases to a steady level for the next few days. Estrogen enhances the effects of progesterone. It takes about 7 days for an egg to travel through the fallopian tube from the ovary to the uterus, and it must be fertilized while in the fallopian tube. If no fertilized egg is implanted in the uterus, the corpus luteum degenerates and the levels of estrogen and progesterone decrease. The endometrium begins to degenerate as the progesterone levels drop, initiating the next menstrual cycle. The decrease in progesterone also allows the hypothalamus of the brain to send GnRH to the anterior pituitary, releasing FSH and LH and starting the cycles again. So this is called menstrual cycle. If fertilization takes place, the egg production or egg release is blocked. If fertilization does not take place, egg release preparation starts again, and this process continues. If a fertilized egg implants in the endometrial lining of the uterine wall, the embryo produces a hormone called human chorionic gonadotropin (HCG) that maintains the corpus luteum. It is this HCG that is measured in the urine during pregnancy tests, during PT tests using the pregnancy test strip. So, if a fertilized egg implants in the endometrial lining, the uterine wall, the embryo produces a hormone called human chorionic gonadotropin. Take note, HCG, small letter H, capital CG, that maintains the corpus luteum. The ovary continues to produce progesterone at high levels, and the menstrual cycle is arrested or stopped for the duration of the pregnancy. Because HCG is unique to pregnancy, it is the hormone detected by pregnancy tests. The figure below usually compares the ovarian and uterine cycles as well as the hormone levels controlling these cycles. So you see the ovarian cycle shown clearly, and you see at day 14 ovulation highest, and we see the levels of FSH and LH. In the other, we see the level of estrogen and progesterone. So these are the days of the menstrual cycle. Menstruation can take place in 22 days. The menstrual cycle can run for 22 days. The menstrual cycle can run for 32 days. So it's a range between 22 to 32 days. But the average is 28 days. So it is 28 days plus or minus in some cases, plus or minus 28 days, plus or minus 4, or plus or minus 6, or plus or minus 7, or plus or minus, I'm not sure it's up to 7, or plus or minus 1, 2, 3, 4, 5, 6. So when the menstrual cycle hangs or is shown to project 28 days in some people, 27 days in some people. So those are 27 days. Every 27 days, their menstruals, they have menstruation. In some people, it's 29 days. In some people, it's 30 days. Sometimes, due to hormonal imbalance or some changes in habits, the menstrual cycle may just change a bit. So people that experience that, doctors have told us they should not be bothered, they should be relaxed, because if those changes that occurs are dealt with and life gets back to normal for them, the normal routine, the menstrual cycle will also get back to its normal state. Now, we want to talk about the male reproductive anatomy. How does the reproductive structure of the male, how does it look like? What does it entail? We have seen that of the female. Now we want to look at that of the male. In the male reproductive system, the scrotum houses the testicles or testes. The singular is testis, the plural is testicles, which produce sperm and some reproductive hormones. Sperm become immobile when kept at body temperature. Therefore, the scrotum and penis are external to the body. The scrotum and penis are external to the body, as shown below. Why is it so? The reason for this is so that a proper temperature is maintained for motility. In land mammals, the pair of testes must be suspended outside the body at about 2 degrees Celsius lower than body temperature to produce viable sperm. Infertility can occur in land mammals when the testes do not descend through the abdominal cavity during fetal development. So the temperature of the testis that produces a sperm must be lower by 2 degrees Celsius than the body temperature, because when sperm is at body temperature, it will not be able to move, and sperm must be able to move to reach the egg to fertilize it. When it gets into the vagina, it must move up, and the movement is not down, it's up. And that's why millions of sperm are produced. Anytime you see that very large number of a particular thing is produced, is because there's a risk of a loss. There's a risk of a loss. The risk of a loss is high. Every 28 days, only one egg is produced by the female, is released. But in the case of a male, anytime he ejaculates, millions and millions of sperm cells are inside of the sperm. The reason is because the sperm is fragile, so it can die easily. If it does not die and the sperm is produced and it's alive but it's not moving, it will not be able to move up the vaginal wall and the uterine wall to fertilize the egg. Though sperm must be produced and stored at temperatures lower than body temperature in the testes, sperm are warmed to body temperature when deposited in the female reproductive tract. The immediate warming of sperm causes them to experience a burst of swimming activity, but then they begin to lose motility after several hours in body temperature. Sperm are produced in the seminiferous tubules inside the testes. The sperm cell production is mediated by 2 different types of cells, which are called nurse-made cells, called Sertoli cells. Sertoli is Sertoli, capital S, which protect the germ cells and promote their development. The second type of cell is the cells of Leydig, Leydig, capital L. These cells of Leydig produce high levels of testosterone once the male reaches adolescence and regulates sperm development. When the sperm have developed flagella, and are nearly mature, they leave the testicles and enter the epididymis, where sperm mature. During ejaculation, the sperm leave the epididymis and enter the vas deferens, which carries the sperm behind the bladder and forms the ejaculatory duct with the duct from the seminal vesicles. Semen is a mixture of sperm and spermatic duct secretions and fluids from accessory glands that contribute most of the semen's volume. The bulk of the semen comes from the accessory glands associated with the male reproductive system. These are the seminal vesicles, the prostate gland, and the bulbourethral gland, all of which are illustrated above and even now. The seminal vesicles, what are they? The seminal vesicles are a pair of glands that make thick, yellowish, and alkaline solution. As sperm are only motile in an alkaline environment, a basic pH is important to reverse the acidity of the vaginal environment. Remember, the vagina is acidic. The solution also contains mucus, fructose, a source of energy for the sperm cells, a coagulating enzyme, ascorbic acid, vitamin C, and local acting hormones called prostaglandins may help stimulate smooth muscle contractions in the uterus. The seminal vesicle glands account for 60% of the bulk of the semen. So what accounts for 60% of the bulk of the semen is the seminal vesicles. What are prostate glands? What do they do? The prostate gland surrounds the urethra, the connection to the urinary bladder. It has a series of short ducts that directly connect to the urethra. The gland is a mixture of smooth muscle and glandular tissue. The muscle provides much of the force needed for ejaculation to occur. The glandular tissue makes a thin, milky fluid that contains citrates, stimulates sperm motility, enzymes, and prostate-specific antigen (PSA). Prostate-specific antigen is a proteolytic enzyme that helps to liquefy the ejaculate several minutes after release from the male. Prostate gland secretions account for about 30% of the bulk of the semen. So the seminal vesicle and its contents 60% of the bulk of the semen. The prostate accounts for about 30% of the bulk of the semen. Now, what accounts for the 10%? Let us go on then. The bulbourethral gland. What is that? What is bulbourethral gland? The bulbourethral gland releases its secretions before the release of the bulk of the semen. The mucous secretions of this gland helps lubricate and neutralize any acid residue in the urethra left over from urine. This usually accounts for a couple of drops of fluid in the total ejaculate and may contain a few sperm. Withdrawal of the penis from the vagina before ejaculation to prevent pregnancy may not work if sperm are present in the bulbourethral gland secretions. The table below briefly summarizes the major organs, locations, and functions of the mammalian male reproductive anatomy. The first organ is the scrotum and is located outside in the external. It carries and supports the testes. Then we have the penis. It's also located external to the body. It delivers urine, it's the copulating organ. Then we have the testis. It's also external. It's also located externally. It produces sperm and male hormones. The testis does not just produce sperm, it also produces male hormones. Let's take note of that. In the post-primary school, we are told that testes produce sperm. We don't want to bug the students too much, so we stop. We don't want to bug those students too much, so we stop at that point with them. But now that you are in the university, we have to let you know that testes do not just produce sperm, it also produces the male hormones. Then, what does the seminal vesicle do? Seminal vesicle contributes to semen production and is located internally. What does the prostate gland do? Prostate gland contributes to semen production and is located internally. What do the bulbourethral glands do? Bulbourethral glands clean urethra at ejaculation. Urethra may contain some remains of urine and be acidic, because urine is acidic. So the bulbourethral gland comes around to make it a sort of alkaline, to clean it up, so that the urine and the sperm does not mix up. So it first comes to clean it up, and it's located in the inside of the organism internally. Spermatogenesis is the next thing we want to look at now. That is the male gametogenesis. Spermatogenesis is illustrated below, and it occurs in the seminiferous tubules in the testes. Stem cells, called spermatogonia, are present at birth but are inactive until puberty. when hormones from the anterior pituitary cause the activation of these cells and the continuous production of sperm. Sperm production continues into old age. To produce sperm, a cell called a spermatocyte, which is a precursor to sperm, undergoes meiosis to produce 4 haploid spermatids. Those spermatids are immature sperm. Once the spermatid develops a flagellum, what is flagellum used for? What is cilia used for? What is leg used for? Can you tell me? Yeah, you can. Leg is used for movement. Legs are used for movement, cilia are used for movement, and also the flagellum is for movement. So once the spermatid develops a flagellum, a tail that allows it to swim, it is called a sperm cell. 4 sperm cells result from each spermatocyte that goes through meiosis. So the diagram is shown here. You have the spermatogonium with 2 N. At mitosis, we have 2 N still, and at meiosis 1, we now have 1 N after meiosis 1. After meiosis 2, we still have 1 N. So after meiosis 1, we have our secondary spermatocyte, and then we have the spermatid produced, there are 4. Then we have differentiation, and then we have the tail, the flagellum with it, and then we have what is the sperm cell produced, and then on it goes in its movement towards the egg inside of around the uterus area. Now, how does hormones control spermatogenesis? That means, how does hormone control the production of sperm? Just like we have in oogenesis, spermatogenesis is controlled by FSH, LH, and LH. Testosterone also play a role in spermatogenesis. Remember, in the female hormonal control of egg production, that's oogenesis, we have FSH, LH, and then we have the progesterone and the other hormone, that's progesterone and the other hormone that is in the female. We have the progesterone and the other hormone in the female. Can you tell me what is the other hormone in the female is? Can you tell me what the other hormone in the female is? We have the progesterone in the female, and we have the estrogen. You have the estrogen in the female also, and they have their different roles to play in the production of the egg and in maintaining the female secondary sexual characteristics. So in the female, we have the FSH, LH, estrogen, and progesterone playing their different roles. In the male, we have the FSH, LH, and we have the testosterone this time playing its own role. So FSH, what does FSH do? It stimulates spermatogenesis in the testes. What does the LH do? It stimulates testosterone production. Now, what does testosterone do? Testosterone further stimulates spermatogenesis. It is also the hormone responsible for the secondary sexual characteristics that develop in the male during adolescence, including a deepening of the voice, the growth of facial, axillary, and pubic hair, and the beginnings of the sex drive. While this doesn't occur in a monthly cycle as in females, the hormones do interact in a feedback cycle which initiates during puberty. In response to signals from the hypothalamus that begin at the onset of puberty in males, the pituitary gland produces FSH. FSH enters the testes to begin facilitating spermatogenesis, which is the production of sperm cells. That the sperm cells, that's the gametes. by meiosis. LH, made by the pituitary gland, also enters the testes to stimulate the production and release of testosterone into the blood. Testosterone stimulates somatogenesis as well as the development of male secondary sex characteristics that include a deepening of the voice, the growth of facial, axillary, and pubic hair, and the beginnings of the sex drive. A negative feedback system occurs in the male when sperm counts get too high, over about 20 million per male. Rising testosterone levels cause Sertoli cells to release the hormone inhibin, which acts on the hypothalamus and pituitary gland to inhibit the release of FSH and LH. The inhibition causes somatogenesis to slow down until proper levels are again achieved. Once the sperm levels are reduced, the Sertoli cells stop releasing inhibin and the sperm count increases. You see, the way nature regulates our body is surprising. It's just like the way nature regulates rain. At a time the rain is falling, and as the rain is falling, you see that the ground is ready to take up the water. You can't see rain just falling and then flooding the soil, the land, except it can flood the streets where there are no drainages. But normally when there are drainages and there are other ways, like in a farmland, in the bush areas, the ground is ready and happy to absorb the water. After absorbing the water, a time of dry season comes. And then even during dry season, in the night before the morning, there is high humidity and there's dew. By the time you wake up in the morning, you find objects placed outside that water has dropped on them. This water gets back to the atmosphere through evaporation. The same thing happens after rain falls, then sunshine. There's a saying in English that after the rain comes the what? Comes the sunshine. So when rain falls, the next thing that will come is the sunshine. And when the sun is very hot and very hot and very hot during the rainy season, just note that a rain is about to fall. So it's a way to balance things up. So you can see also in the sperm formation, when there's excess of sperm, the body regulates. So the Sertoli cells will release inhibin, which will now prevent the further production of too many sperms. So the sperm production reduces. But when the sperm level is low, this same Sertoli will now lift up his hand and will stop inhibiting and will stop releasing inhibin, and then the sperm count will increase again. The sperm count will increase again when there is low levels of sperm. So this is a way for the body to regulate in itself. How are gametes made? And when are the gametes made? When are the gametes made and how are they made? That is what we want to talk about now. That is what we want to talk about now. That is what we want to talk about now. When gametes start to form, that is, egg production begins during embryonic development, that is before birth. Then it is arrested during meiosis until puberty. Sperm production does not begin until puberty. When gametes finish being made, egg production is not actually completed until after fertilization, while sperm production is complete just before ejaculation. How many gametes are made from a gamete stem cell? Egg production results in only a single egg. From each egg stem cell, sperm production results in 4 sperm 4 sperm from each sperm stem cell. Rate of production of these gametes. Once an individual enters puberty, sperm production is continuous in a conveyor belt process. Sperm production is continuous, it keeps flowing, but egg production occurs one at a time at each menstrual cycle, and it gets to a point in the life of a man, sperm production does not stop, it continues till the man dies, except there are some special issues with health. But in the case of egg production, the female is estimated to produce 330 eggs during her lifetime. So the earlier a female starts menstruation, the earlier she's going to stop menstruation, and that is what they call menopause. You can read more about that and drop in the comment section below and answer the question, how many eggs does a female produce during her lifetime? How many eggs does a female produce during her lifetime? I have said it's about 330 eggs, so you can calculate how many years she's going to produce the egg. So you are going to give me your own answer for the estimated number of eggs a female will produce during her lifetime. And you don't just give me the number and just write, as I said, 330. You have to give me the source. Where did you find it? Where did you find the answer that it is 328 or 330 or 340? You have to write it. Different materials are going to give you different answers, because it is not exact. A particular female may menstruate for a number of years, X number of years. Another female may menstruate for Y number of years. Another female may menstruate for Z number of years. That means a menstrual cycle may go on before she reaches menopause. I'm not saying that menstruation continues without stop, as this is bleeding. No. What I mean is that the menstrual cycle, how many times is she going to produce egg? Because during the menstrual cycle, it is egg that is produced, that is not fertilized, that drops. So you calculate all that, the number of babies and the number of times. Usually, the female should menstruate 12 times in a year or thereabout. But in abnormal cases, it is below or above by 1 or by 2. So let's read more on that and drop what you find out in the comment section below. We have to also do something. It's not everything that the teacher or lecturer is going to be telling you, or that the instructor is going to be saying to you and telling you. You also have to find out something. In fact, the one you find out on your own, you see that you hold it is held better than the one that the lecturer or teacher or instructor just tells you. What is the rate of production of this sperm? Once an individual enters puberty, sperm production is continuous in a conveyor belt process. Egg production occurs one at a time at each menstrual cycle. Now, before we call it a day with this subject of reproduction in mammals or in animals, we want to talk about contraception and birth control. Contraception and birth control is part of the content of this material at this time that I want to talk about. Since we talk about reproduction, it is very, very important that we talk about contraception and birth control also. And we are going to do that after this short break. Yes, welcome back, dear students. As I said earlier, we want to talk about contraception and birth control, how we can control birth for those who are married and don't want to have children yet. They can control, or those who are married and they plan to have 3 or 4 children or 1 or 2 children, and they've had those children and they want to see and they don't want to have more children. That is what we mean by birth control. Now, methods of contraception to prevent pregnancy have many probabilities of success and also probability of failure. In the diagram that is shown currently scrolling, the failure rate is given as the percent of women who become pregnant during the first year of use of that method. So the diagram is showing the effectiveness of different contraceptive methods. You have to take your time and look at the diagram properly so that you can be able to get what it's saying. We have the implant. The failure rate for implant is 0.05%. Intrauterine device IUD, the failure rate is 0.2% and up to 0.8%. For sterilization in males, the failure rate is 0.15%, while in female is 0.5%. And we have the use of needles, injectable, the failure rate is 6%. For pills that women take, the failure rate is 9%. For patch, the failure rate is 9%. And for ring, the failure rate is 9%. The use of diaphragm in women, the failure rate is 12%. Then the use of condom, the failure rate is 18%. The use of female condom, the failure rate is 21%. The withdrawal method, the failure rate is 22%, while the sponge method, the failure rate is 24%. The fertility awareness-based method, the failure rate is 24%. And then the use of the spermicide, the failure rate is 28%. So these are the different failure rates of different birth control methods. So that will help you to know that no birth control or contraceptive is 100% sure to prevent pregnancy. Barrier method is one, and it includes the use of condom, cervical caps, and diaphragms. It blocks sperm from entering the uterus, preventing fertilization. Spermicides are chemicals that are placed in the vagina that kill sperm. Sponges, which are saturated with spermicides, are placed in the vagina at the cervical opening. Combinations of spermicidal chemicals and barrier methods achieve lower failure rates than do the methods when used separately. Natural family planning is based on the monitoring of the menstrual cycle and having intercourse only during times when the egg is not present. A woman's body temperature may rise a degree Celsius at ovulation, and the cervical mucus may increase in volume and become more pliable. These changes give a general indication of when intercourse is more or less likely to result in fertilization. Withdrawal involves the removal of the penis from the vagina during intercourse before ejaculation occurs. This method, which has a high failure rate due to the possible presence of sperm in the bulbourethral gland secretion, which may enter the vagina prior to removing the penis. Hormonal methods use synthetic progesterone, sometimes in combination with estrogen, to inhibit the hypothalamus from releasing FSH or LH, and thus prevent an egg from being available for fertilization. This method of administering the hormone affects failure rate. The most reliable method with a failure rate of less than 1% is implantation of the hormone under the skin. The same rate can be achieved through using an intrauterine device, IUD. IUDs are inserted into the uterus and establish an inflammatory condition that prevents fertilized eggs from implanting into the uterine wall. Some IUDs also release progesterone. Emergency contraception, also known as Plan B, is also a hormone-based method of contraception. One common misconception about emergency contraception is that it prevents implantation after fertilization. However, like other contraceptive methods, it does not induce abortion. It has no impact after fertilization. During a vasectomy, a section of the vas deferens is removed, preventing sperm from being passed out of the body during ejaculation and preventing fertilization. The equivalent process in women is called a tubal ligation. It is analogous to a vasectomy in males in that the oviducts are severed and sealed. Tubal ligation and vasectomy are considered permanent prevention, while other methods are reversible and provide short-term contraception. You have to think about the most effective strategies for preventing pregnancy. Methods in combination, such as pharmaceutical chemicals and barrier, prevent pregnancy more effective than do the methods when used separately. What causes failure of this contraceptive or of this birth control methods? Some sperm are present in pre-ejaculate secretions, so without a barrier, some sperm may still enter the vagina. Hormonal methods vary in success by hormone delivery method, a method not pictured in emergency contraception, also known as Plan B. This hormone-based method of contraception works against the egg and the sperm simultaneously and before the fertilization occurs. So I would like you to read more on birth control methods and on other areas that we have spoken about. So we are talking about reproduction in animals, reproduction in mammals, and we have seen differences that occur. We've seen the female reproductive organ and reproductive system, and we have also seen that of the male. So I want you to take your time and go over everything we've done and read through, and you take this assignment and you tell me the function of the ovary. You also tell me the functions of the oviducts and the other name for the oviduct and the other name for uterus, and the function of the uterus, the function of the oviduct, the function of the ovary, and the function of the cervix. And you also tell me the function of the vagina. Tell me the so that's sufficient in the female. In the male, you will tell me the function of the testes, the function of the seminiferous tubus, the function of the prostate gland, the function of the urethra, and the function of the scrotum, also the function of the epididymis and the function of the vas deferens. And I think that also should be done, should be okay for the male. So you are going to get for me a diagram of the egg and also a diagram of a sperm, of a spermatozoa. You draw them and you label them and your material. So, and then you make a table and give me differences between the sperm and the ovum. You give me differences between the sperm and the ovum. Also, you draw a table and you give me similarities between the sperm and the ovum, if there's any, and there should be. So you give me the similarities. Most of the time we talk about the differences alone, and we don't bother our head with the similarities. So in function, they have similarities. In physical appearance, they may not have any similarity, but in functionally, they have similarities. So I'm going to talk to you some other time. Please take your time, go over this material, and we are going to meet again in happiness and enjoy in another subject matter. Please take your time to go over these materials again. I'm saying it again. There's this method of studying called the SQ3R study technique. SQ3R study technique. There are different types of study techniques in the world, but the SQ3R is the most advanced and is the best. The S in SQ3R stands for survey. The Q stands for question. The first R stands for read. The second R stands for recite. The third R stands for recall. So the first thing you do when you want to study is that you survey. You look through the material from beginning to middle to the end. Then the next thing is to question. Then you ask yourself questions. You look at the subheadings in the topic. You ask yourself, you can write the question down, you can have it in your mind. It's best to write it down. Then the first R is to read. As you're reading, you are looking for answers to those questions that you've written down. Then as you've gotten the answers, the next thing you do is to recite. You go back to the question and then you recite the answer. You go back to the question, you recite the answer without looking at the material. Then you now review. You now look at it. What did you know before you started the learning, and what do you know when you completed the learning? Then you repeat that process. Before you know it, you are going to become a guru. Bye for now.